All right, welcome back to the second video in chapter 24. So now we're going to talk about the difference between aerobic and anaerobic respiration. When we last talked in the previous video, I said this video we were going to start out talking about pyruvate's fate, or the fate of pyruvate. This fate is dependent on if oxygen is available or not. So if oxygen is available, then we're going to go through an aerobic process. But if oxygen is not available, then we move into an anaerobic process. So aerobic means with oxygen, and aerobic means without oxygen. And so if there's oxygen available, glucose is going to get converted into pyruvate, and then the pyruvate moves into the next step of aerobic respiration, which occurs in the mitochondria. The NADH and the pyruvate are going to pass through the plasma membrane of the mitochondria, and we're going to go into the next stage. That's with oxygen. If oxygen is not available, then when glucose is converted to pyruvate, the pyruvate is then reduced to lactate. So we oxidize the glucose in the first part of glycolysis, and then when glycolysis ends, we then move into a process known as fermentation, and we reduce the pyruvate. This reduction of pyruvate allows us to reproduce NAD from the NADH. We need NAD so that we can oxidize more glucose. NAD is the oxidizing agent. Glucose would be the reducing agent. We also are decreasing the amount of pyruvate in the cell. If there's too much pyruvate available, then pyruvate will actually bind to the enzymes in the glycolytic pathway and stop that pathway from running. And then you would not be able to produce any ATP. So lactate is a byproduct of anaerobic fermentation. And when you have a lot of lactate in your system, it actually causes problems like muscle aches. It can cause your muscles to stop functioning properly. Think about when you've had an illness and your body started to ache. Part of that, depending on what the illness was, could be because you are trying to produce more energy than you have oxygen available, and so you're producing lactate as a byproduct, which causes muscle soreness. So the next step, your textbook doesn't really give it a big name. It says transformation of pyruvate to acetyl-CoA. I give it a name called transition. This process is an oxidative decarboxylation process. So we are going to oxidize the pyruvate, but we're also going to remove a carbon from it. So this is the first step where we're going to have oxygen needed. And this occurs in the matrix of the mitochondria. So once we enter the mitochondria, we now need oxygen to be available. So we take the pyruvate, and each of the pyruvates, we'll go through this. We're going to remove a carbon and add it to the oxygen that's available. And then we're going to mix the pyruvate with the coenzyme A that is there. We oxidize that pyruvate and produce an acetyl group carried on coenzyme A. We oxidize it using NAD again. So NADH is formed. Because we have two pyruvates, we're going to produce 2 carbon dioxide waste products, 2 NADH, and 2 acetyl coenzyme A molecules. The next step is the Krebs cycle, or citric acid cycle. It's named the Krebs cycle because the individual who found it was named Krebs, identified the cycle, and the citric acid cycle for what it produces. So in this cycle, it is a cyclic pathway, which means we start and we end with the same molecule, this 4-carbon oxaloacetate. The acetyl group, which is in the matrix already because it was just produced, acetyl-CoA, moves to the citric acid cycle, which is also in the matrix of the mitochondria. The coenzyme A drops off the acetyl group. The acetyl group binds to the oxaloacetate, and we produce citrate. Citrate is then, through multiple enzymatic reactions, converted to different molecules. It's oxidized, and it releases all of the hydrogens that were in the original acetyl group. So in the acetyl group, we had 2 carbons. So in this process, we're going to decarboxylate the entire acetyl group, so there will be no carbons left. We're also going to oxidize the acetyl molecule, the acetyl group, completely, so there's no hydrogens or electrons available. And in that process, we are converting the citrate into multiple new molecules, which you don't have to know these names, but you do have to know is that once citrate is produced, we decarboxylate it completely, we oxidize it completely back to the oxaloacetate that started the process. And in this process, we produce a small amount of ATP through a process called substrate level phosphorylation. This process does occur twice. So in the end, we're going to produce a total of 4 carbon dioxides, and I'm going to go back just so I can show you. We're going to produce a total of 6 NADH, so there's 1, 2, and 3, but this is just for one acetyl group, so remember this occurs twice. And then we're going to produce 1 FADH right here. FAD, flavin adenine dinucleotide, is another coenzyme that is an oxidizing agent. So it's going to pick up those hydrogens as we remove them from the original acetyl that came in. So we produce a total of 2 FADH, 6 NADH, and 2 ATP in this process. The last step of the entire process is known as the electron transport chain. And in this process, we use oxygen to help us oxidize the NADH and FADH back to their NAD and FAD. And we're going to produce a lot of ATP in a process known as oxidative phosphorylation. So here you can see NADH coming in, and there's FADH. We have a total of 10 NADH, 2 from the glycolytic pathway, 2 from transition, and 6 from citric acid cycle that are going to come in, and they're going to get oxidized by the oxygen that's there. And they're going to, when they get oxidized, they remove the hydrogen ions and the electrons. The electrons are passed from one protein carrier to another until they attach to oxygen. The hydrogen ions are then going to be pumped across the membrane into the intermembrane space, which has a higher concentration of hydrogen ions, but the electrons moving, that shuffling of electrons, is what energizes these proteins to allow hydrogens to move across. So we end up with this high energy concentration up here. FADH also gets oxidized, but it drops its proteins off at the second pump, or at the second protein. And because of that, there's less energy that's going to actually be produced. So we have hydrogens going through here, hydrogen ions going through here, and we have this high concentration of hydrogen ions in the intermembrane space. Whenever you have a high concentration of any molecule on one side of a membrane and lower on the other, they want to move from where they are to where they aren't. So what happens is they have one route to move. They can move through this large enzyme over here, but this enzyme is known as ATP synthase. So when the hydrogen ions flow across, it spins the complex and phosphates are added to the ADP, producing ATP. So this is that oxidative, we're oxidizing the NADH and FADH. Phosphorylation, we're adding the phosphates to the ADP, producing ATP. All right, this is what I just talked about. All the NADH drop their electrons and hydrogens at the first protein. All FADH drop their hydrogens and electrons off at the second proteins. Hydrogens are shuttled across the matrix into the intermembrane space. Electrons move from one carrier to the next carrier, and that drives the movement of hydrogen from the matrix to the intermembrane space. Hydrogen ions then move back across from the highly concentrated intermembrane space through the ATP synthase molecule back into the matrix, where they can attach to the oxygen. For every NADH that drops off its hydrogens, we produce 3 ATP. And for every FADH that drops off its hydrogens, we produce 2 ATP. The hydrogens and electrons, once they move back into the matrix, they're going to attach to oxygen, and we're going to produce water as a byproduct of cell respiration. Now, I talked about the amount of NADH produced. So you have 2 here, and 6 here. So what that means is there's 10 NADH. Because for each NADH we produce 3 ATP, we're going to have a total of 30 ATP produced in the electron transport chain. And then I said that for every FADH we produce 2 ATP. We produce 2 FADH here in the citric acid cycle. 2 times 2 is 4. So at the end we produce a total of 34 ATP from the electron transport chain. Now we produce 2 ATP up here, and we produce 2 ATP here. So that's 2 plus 2 plus 34 is 38 ATP. But to move molecules across the plasma membrane of the mitochondria, it costs energy. So we lose some ATP, a total of 2 ATP in that process. And so we end up with a net gain of 36 ATP for the breakdown of one glucose molecule. We can use other molecules to produce energy. And one example is fatty acids. So we can break fatty acids down through a process known as beta oxidation. Fatty acids are not our preferred energy, but they are energy that our cells will use. And so we break fatty acids down by a process known as beta oxidation, forming small groups called acetyl groups. These acetyl groups are then going to move directly into the Krebs cycle on coenzyme A. And so this process is only used during aerobic process, or is an only aerobic process. We can also use proteins. So when we break down proteins, we form different amino acids, and these different amino acids can enter in different areas of the aerobic respiration. Some enter into the glycolytic pathway, and some are going to move directly into the Krebs cycle or citric acid cycle. But before we can do that, we have to first remove this amine portion. So the removal of an amine portion is known as deamination. So we deaminate the molecule, and when we do that, then we have this protein that has, so you have the remainder group, you have the reactive group, I should say. We have that central carbon ring, and we have that C double bond OOH, or that carboxylic acid group. And then we have this NH3 that's separated, okay? Then this here can move into those different processes. So we are skipping gluconeogenesis. I've talked about that in chapter 17 when we talked about cortisol and the stress response. We're going to skip lipogenesis, the urea cycle, metabolic states of the body, energy and heat balance, and the nutrition portion. Each of these small sections you can read on your own to get that information. I'm skipping them so that we can get through and get into the urinary system, because we still have a lot of material and we only have a few more weeks left. So with that, we are done with the chapter, so super short chapter. I will post these and pull them through Ed Puzzle. Have a great day. Bye.